Good evening, everybody, and welcome to the latest installment of our EcoFlora virtual seminar series. My name is Lydia Paradiso, and I'm a doctoral student at the New York Botanical Garden and a graduate fellow working with the New York City EcoFlora project. EcoFlora was first started at NYBG back in 2017 with a grant from the Institute of Museum and Library Services. EcoFlora is a community science project with three main goals: to investigate urban ecosystems and urbanization, to support open source biodiversity data, and to increase understanding and appreciation of plant life. To date, over 30,000 observers have made almost 900,000 observations of plants and their biotic partners in New York City and posted them to iNaturalist. For reference, about 60% of those 900,000 are of plants and fungi, while the remaining 40% are organisms like insects and animals that are pollinators, seed dispersers, and more. You can explore these observations on our project page on iNaturalist, which I'm going to link in the chat right now. Each month, we also sponsor an EcoQuest challenge, which encourages New Yorkers to observe a particular species, group of plants, or interaction, and post them on iNaturalist. For March, our EcoQuest is entitled Wings in Spring. Insects form the biological foundation in which our ecosystems rely. Many species that have overwintered emerge at the early beginning of spring, such as the widely observed and long-lived morning cloak butterfly, as well as many other winged insects, including moths, bees, wasps, flies, beetles, and winged winter ants. The queens of many bee species also emerge in March, looking for a suitable place for a new home, while others get busy pollinating the early blooms of maple trees. So far in this month, we found 38 species of insects that have been observed out and about in the city, many even caught in the act of pollination. Those with most sightings include the American winter ant and the European honeybee. You can check out all of these observations on the iNaturalist page for that project. We're also planning a field trip to complement that EcoQuest for this coming Sunday, the 26th in Brooklyn. The exact location is still to be determined, so stay tuned to your inboxes for more information. I'm going to post a link to join our mailing list if you are not already on it. We also have some quick announcements before we go into our talk for today. The two next talks that we have scheduled, I just wanted to give some information about those. On April 11th, a Tuesday, we're going to be hosting Clara Pregitzer from the Natural Areas Conservancy, who will talk about carbon storage in urban forests. And in May, on Thursday, May 4th, we'll have a talk by George Lazewski from the Urban Soil Institute, who's going to talk to us about different soil types that can be found in the city, their role in our ecosystems, and work that's being done to understand and conserve them. So I'm going to just post both of the links for both of those Zoom registrations into the chat, but we will also send out more information via email. So if you are not already on our mailing list and you'd like to sign up, we got another link in here that is that sign up for our mailing list. So with those little notes, I'm going to turn it over to Tomi to introduce our speaker for tonight.
Hello, everybody. I hope you have been enjoying the first bits of spring in the warm sun. I'm going to start off this seminar with a land acknowledgement, per usual. So I'd like to start this seminar with a land acknowledgement by recognizing the original people of this land, the Lenape people, as well as their deep connection to the Lenape home king. Well, oh my gosh, this is just a tongue twister always. The Lenape-Hoking homeland. As an organization dedicated to advocating for the more than human world, we must acknowledge those who are original stewards of the land. We believe in the importance of acknowledging the settler colonial genocide perpetrated against the indigenous communities, as well as the resilience of the Lenape, who still today continue to resist erasure. Awesome. Okay, so for our speaker tonight is Heather Liljengren, the Supervising Seed Collector and Field Taxonomist for the New York City Parks Greenbelt Native Plant Center located in Staten Island. Ms. Liljengren actively manages the seed collection and banking programs at the 13-acre greenhouse, nursery, and seed bank complex, which provides local genetic source material for restoration projects throughout New York City. For 10 years, she was an advisor to the Mid-Atlantic Regional Seed Bank, a cooperative conservation effort to collect seeds from targeted native plant species across every ecoregion in the Mid-Atlantic. As a public speaker and teacher throughout the New York City Metro Area, Ms. Liljengren promotes the importance of using locally adapted native plant species in every reach of restoration and functional landscape design. In her spare time, she is busy with her family, fostering the love for all things green with her 2 children and listening to her husband butcher the Latin names of all of those green things. Tonight, Heather will tell us more about her work as a seed collector. Please feel free to enter any questions into the comments or comments into the chat as we go. At the conclusion of the talk, we will go through as many questions as we are able. If you have a question or comment you would like to ask verbally at the end of the talk, you can raise your hand or use the raise hand feature or note in the chat. We'll have the opportunity to do so. Also, if you are unable to stay for the whole talk or would like to share it with others, a recording will be posted to the New York Botanic Gardens YouTube channel in the EcoFlora playlist. All right, without further ado, I will turn it over to Heather.
Hi, good evening. Thank you all for coming. I know it's a tough time in the middle of dinner, but I'm glad you're here. Doing this talk makes me very excited for spring and the field season that's upon us. So I'm so happy to be here. I'm going to share my screen and get right to this. Oh gosh. Okay. So urban genetic pools, the reservoirs of adaptive.
I don't think it's sharing.
I don't think it actually shared yet.
Oh.
Okay.
Did it share yet?
I haven't seen anything come up.
Okay, let me try this again.
There we go.
Yeah.
There we go. Okay. So let me start the slideshow. Okay.
Perfect.
So urban genetic pools, the reservoirs of adaptive potential in the seed collectors who are after it. So I love to talk about the dichotomy of our urban areas, trash and beauty. Both of these photos were taken in New York City. And it's the reality that we live in a very highly populated area. The picture on the left is unfortunately what happens to a lot of our waterways. They're littered with trash. But the picture on the right is, a beautiful spot on Hunter Island in the Bronx where you just forget that you're in one of the most highly populated places in the world. And this is the reality that we live in. And both of them are important and both of them are adding to the incredible genetic diversity that we have here within New York City. So unfortunately, this is how a lot of our spaces look. I travel throughout the 5 boroughs, going to every reach of the natural areas, and a lot of them become trash heaps. And they really endure these harsh conditions and pollution, but our flora continues to exist. And our flora continues to thrive even amongst all of this fragmentation and pollution. And it's because it is resilient. It's a buzzword, but it is something that I have seen throughout the years. Our plants have figured out how to live amongst everything that we have thrown at it. And it really is a true testament to the stronghold that they have here in the landscape. This is some Spartina alterniflora, our marsh cord grass growing out of an old boat that washed up. This is the same species growing out of an old motor. And if you could see the mussels that are also colonizing it. This is a Carrick strictus swamp out in Staten Island in Wolf's Pond. It's right on the side of a road. You could literally walk off the road and come into this beautiful swamp with, of course, some garbage in the way, but this swamp, it provides a refuge for so many of the important wildlife species that live there. This is some of the Canada Mayflower, Myanthemum canadense, growing out of some rusty old pieces of metal in the middle of the woods. There's a lot of burnt out cars all over. There was a lot of insurance fraud that happened in the 70s and 80s throughout New York City's urban spaces, and they still exist, and the plants have figured out how to just grow amongst it. Our plants find these little cracks everywhere along the sidewalks and pathways that, and they become a great home for them. This is our native viola, Viola sororia, the common blue violet. This is some osmunda, our cinnamon ferns that will grow amongst tires and some old cars. And this has got to be one of my favorite places to go and find spontaneous flora. This is the Metro North trestles up on the Upper East Side. If you walk between 101st Street and 105th Street on Park Avenue, you can find these, ferns just growing right alongside the walls there. It is magnificent. If you ever get a chance to go check it out, please do so. This is Woodsia obtusa, the blunt cliff fern, blunt-lobed cliff fern. And they just have found their niche and they've figured out how to survive here. And that's important, and it should be a lesson that we continue to follow and then try to capture the seeds of these species. And we're seeing plant species adapt in real time. On the left here, you can see the Solidago sempervirens out there in all its glory on Rockaway Beach in Queens. This is its natural habitat along the coastline. But as we have changed the landscape and we've put roads, coming off the coastlines, and we're salting all of our roads, you'll see this species pop up all along our highways and roadsides, like here in the Gowanus Canal on the right-hand side. And then this, although it is across the water at Sandy Hook Beach in New Jersey, this is another example of a species that has found its niche. This is the Eastern Red Cedar on the left in all its glory, out there in the middle of the maritime grassland, getting all the nutrients and sun and everything that it needs. But then it's bonsai'd itself on top of the fort here at Fort Hancock. And it's this, contorted little tiny specimen that is pale in comparison to, the species, the specimen on the left, but still so valuable and so important on how it's been able to figure out how to survive and how to continue to exist within these conditions. And this brings up the basic ethos of why the Greenbelt Native Plant Center exists. We are trying to capture the genetic diversity of our local flora, and it's through seed collection that we're able to do that. The genetics that are wrapped up in a seed, they hold that variation and adaptive potential that we can put back out there into the landscape. And there have been plenty of studies done that have shown time and time again why using local adapted plants are better in the long run for survival. And all of these reasons that are listed go to show why this is so important and why we shouldn't just be, just because it says it's a native plant, doesn't mean that it's the right native plant for the right place. We need to understand where our plants are coming from and putting the right native plant in the right place. And this has been going on since 1995, believe it or not. We have come a long way in all those years, but the Greenbelt Native Plant Center has existed in a form since 1995. And our mission is to provide native plants and seeds from local plant populations in support of restoration and management of the city's most valuable natural areas. This is a 13-acre farm, an old family farm out in Staten Island that the city purchased in 1992. And this has been the place where we've been able to continue the work of a couple people that really had a vision on why native plants were so important and so important in a changing urban landscape. And it all starts with our seed collection program. We collect and store seed only from native plant populations. And the 5 boroughs are the most local and the most important, but our collection area spans beyond that. And we actually collect within 25 counties in 3 different states in a 100-mile radius around Columbus Circle. From Columbus Circle. And we're able to do this work and have been able to do this work for so long because we have an internationally recognized seed bank on our property. And if anyone has ever been to our facility, I'm sure you have stepped into the seed bank, and it really is a wonderful thing to experience. People love the smell of it. They love to, look through all the envelopes and bags that we have. And really, this is kind of our gold mine. We've been putting collections into the seed bank since 2006. Before that, we had a very ad hoc way of storing seed, but and even some of that seed that, eventually made its way from being collected in previous years, it still can germinate today. So this is considered a short to mid-term storage seed bank. It's a cool, dry storage. And this is meant to be an active seed bank. We're not doing the Noah's Ark. We're not trying to do any long-term storage here. We're really trying to have this be an active seed bank that is being pulled from continuously to be growing plants. And we have other institutions that also store their seed here as well. But really, this is a city-owned facility, and we are serving the city of New York with this seed bank. We have all of the seed is used for our production. This is if you look up on the upper left-hand corner, this is another cooler that we have. It's essentially our winter that we try to create to help break dormancy of all of this seed. We also have some outdoor beds that we do some sowing in as well, but most of our seed goes through, a cold, moist stratification period to break dormancy. We have a number of greenhouses that we then use for the production. And it really depends, all of our production is mostly project driven. So we are custom growing for particular projects, or we have standing lists of species and plants that projects will want year after year. And that's really it's really what fuels our production. And then everything is in this nursery yard that we have 4 acres of irrigated nursery yard. We have 62 Quonsets that line our property, and this is holding all of our woody plants, our herbaceous plants. And we're a restoration nursery. We're not growing bald and burlap trees. The largest size material that we grow is in 1-gallon containers. There have been some particular projects where we've stepped things up for 2 gallons, and there may have even been some random 5-gallon containers, but pretty much across the board, 1 gallons is the largest. And we also have a deer fence that now protects our property. Deer are pretty rampant in Staten Island, and we were the buffet for a long time. But now with our deer fence, we are able to protect all of our plants. And we also have a seed increase program. So not only are we collecting seed for production at the nursery, but our wild collections go to start the seed are used in the plots for the seed increase program. So the idea is that you take 3 to 5 different collections of one species and you put plugs of those species into one plot. And this is going to put together groups of plants that would have never been able to exchange genes before, because they're from all different locations that would have never been in contact with each other. So we're trying to create this really strong, genetically diverse F1 harvest off of these seed plots to really, have a good seed product to be used for different seeding projects throughout the 5 boroughs. There's been 68 different species that have been tested in our plots. We've done seed mixes that have been generalized, different seed mixes for different types of habitats. And we do a lot of custom mixes. So people will come to us and want to seed a particular area, maybe even just, 1 acre or less than, and we're putting together custom seed mixes for that. And not only do we grow at the property at the nursery, there's also seed increase plots at Floyd Bennett Field and at Decker Farm, which is in Staten Island. And, we're in such a unique part of New York State. When you think about New York State as a whole and you say, well, it's a New York native, that can mean so many different things. Our flora is so different than the flora up in the Adirondacks. And if you look at this map, you can really visually see how unique the area that we live in is compared to the rest of the state. Our flora is more like that of New Jersey, parts of Connecticut, and further south. So we have 3 eco regions that are represented in New York City. We have the Northeast Coastal Zone, the Atlantic Coastal Pine Barrens, and the Northern Piedmont. And this is really the foundation of the habitats that exist in New York City. Granted, you throw in these sort of urbanized ecosystems and how that has altered a lot of our species dynamics, but at the base of it, these are the ecoregions that are represented, and these are the ecotypes. This is what we are considering, that we're collecting for, to try to capture the specific ecotypes of an area, because these are plants that are adapted to particular soils, climates, elevation, precipitation. It's no longer, what's your hardiness zone. It's really all of these things really will inform you on what is the best locally adapted species for your area. And this is our collection range. The New York Metropolitan Flora Project, which was a project started by the Brooklyn Botanic Gardens. This was their maps of what they considered to be the local flora. And they would hike around and document the flora, within these counties. And this is the exact collection range that we use today. So what's cool about this project is that, they were documenting not only the historical data by using herbarium specimens and whatnot, but they were doing a re-inventory to sort of understand the species that the species distribution, after a certain year, so after 1990. So, it really showed how the distribution of species has moved, increased or decreased. And I would start, asking the botanists at with this project, have you seen this species? Can you give me some locations of where this species might be that I'm looking for? And they would print out their huge spreadsheets with all the different locations that they'd find these species in. And then I would just have to go on the hunt. And this was really the basis for me on how I started to understand where species were throughout this region and how to go find them and what their habitats were. And it's a fantastic project. And all of their herbarium specimens are now located at the New York Botanic Gardens, and you can go and see them there. And really, this has been an important part of our story, because always the question is what is native and what is a naturally occurring population? Because we're not collecting from species that have been planted. We're not collecting from restoration sites. We want to know the plant populations that have existed here and continue to exist here and capture the genetics of those populations. So we have used so much data, historical data, to really understand the landscape. And this is a list of all of these different, things that we have used. And it really, we're lucky to live in an area where the flora has been documented for hundreds of years. Botanists, you can, go back into journals and read the species that botanists had seen. The Torrey Botanical Journals are a great thing to dig into. There is this particular one that I wanted to show you. If you look at June 12th from 1966 at Wolf's Pond Park in Staten Island, right at the bottom, it says, along the road was a stand of Tripsacum dactyloides, the gamma grass. That population is still there today. We still collect off of that population. Tripsacum dactyloides is a state listed species. It's an S2, it's threatened. It grows still in Staten Island and in the Bronx. And this one particular population, it's not that large, but it's still there right on the side of the road, exactly where this, botanist wrote about it in their field notes. So things like this have been, really important to bolster, our understanding of the landscape and how to go and find plant populations. This is the field notes preceding iNaturalist. This was a great website that was put together by a bunch of different botanists, and it would just be a list of plants that were in a park. And I would go onto this website and I would look at the park that I was going to go to for that day, and I would have an understanding of what plants I might find. But this is just a observational data. There was not much population data that went along with these observations. And I think even with iNaturalist, it's a shortfall of observational data, because if you were really looking to understand, is it a healthy population of plants? What are the numbers? That's all something that we need to know for seed collection. So it can be the sort of the stepping stone, but really getting out there into the field and assessing a population is a huge part of how we start our job. And the process of seed collection, there are many steps to it, but really, I think that it comes down to a really intimate relationship with these plants and all the species. And, there is a process to understand, how to know when a seed is ready to collect. Is it enough to collect from? And I'm going to bring you through that process. So we do a lot of things from start to finish. Not only are we doing the seed collection, but we also then do all the seed processing, storage, and regeneration at GMPC. We're a closed system, and why we're a bit unique in the industry. So all of this information and all of these steps are done in-house. And so it really helps to be able to understand the species at a level where you are able to talk with the propagator to figure out maybe how to break that dormancy by understanding what's happening with it out in the landscape, who eats that seed, when does it fall on the ground. All of these things are important into understanding how to break dormancy and be able to propagate plants. And we do follow a particular protocol. We are trained in this. It's not something that we just, willy-nilly go out and do. There are international protocols. This is the Seeds of Success. This is our national collection program that was born out of the Millennium Seed Bank Project, which was a worldwide collection program. So there are really particular steps that you need to take when assessing, are you able to collect seed or not? And really it's the population assessment is one of the most important parts. And there's so many questions to ask when you're out there in the field. Every place that we go to collect seed, we have permission, we have permits. And that's important because we want to make sure that we are in good standing with the landowners. And there's a lot of red tape that sometimes that comes along with that. But we are doing this legally. That's number one. And then it's really assessing the population itself. We're not collecting from one plant. We can't do that. That's not going to be capturing the genetic diversity of a population. So there's all these different questions that we need to ask when we are out in the field assessing. And there is, a basic rule of thumb. If you're planning to make seed collections for reproduction, you want to target populations of at least 250 plants in a population, and you're collecting from at least 50 plants, and that's really going to maximize your genetic diversity. But what's important about this is not only it's getting a sample from the population, you're not over collecting. It is so important to make sure that you're leaving enough seed for the natural seed bank and the wildlife that depends on that seed. It's hard sometimes. You want to be greedy when you're out there. If you find, like a really great population, there's a lot of seed and we really need this species, it's hard to kind of dial it back sometimes. But, you don't want to collect more than 20% of the seed that's available on a given day. And seed will mature throughout, a particular window. So if you're there on that day and you're only collecting 20%, you're not going to be harming the population. And over collection is a problem. It's happened throughout the decades and centuries of people over collecting plants and them disappearing from the landscape. So that is never something that we want to do. So we really try hard to, stick to these rules. And when you're doing seed collecting, there's a real basic 3 different ways that you can do it. It could be simple, random, and even. You're just moving throughout the population. You're, grabbing from plants. If you really want to stick to that 20%, in your mind, you're grabbing from every 5th plant you see. You can be systematic about it. You can, if you're collecting with a number of people, if you see on the lower photo, we're moving through a field systematically. Each person's kind of taking a line and you're making sure that you're covering the population. or you're doing a stratified random sampling where you're dividing the populations into smaller groups, and maybe you're only collecting from the like if maybe you're trying to collect from the southern end of the population, the northern, the eastern, and the western. So really you're making sure that you're covering the population, and you want to make sure that you're getting those outliers. You don't want to discriminate on what's the biggest plant, what's the most beautiful, what has the most fruit. Those species that maybe only produce a little bit of seed, they might have a particular adaptation that's important. It may only produce because of a particular lack of nutrient that it was in its place, but if it was able to spread its seed elsewhere, it could have those particular adaptations that are important to continue to increase those numbers. So really it's moving through a population and really trying to make sure that you're getting a good sample. And we use a variety of collection bags. We're just out there in with a backpack with some materials. So, and it depends, it's species dependent on what we're collecting in. So it's either paper envelopes or plastic Ziploc bags for fleshy fruits, cheesecloth bags, saddle bags if we're collecting a lot of tree nuts. So really it depends on, what you're collecting on the type of bag you're using. But what's most important is the field data collection. And this is all the information that we take when we're making a collection about the site itself. So here is our collection form. We still have paper forms, but we do have this form online as well. We do some survey 1, 2, 3 forms, but this is our original paper form. This is all the information we collect when we are making a seed collection. And all of this is housed in a database. So if you come to us to buy plants and you want to know, where that plant was collected and the associated species with it, the soil textures and anything else that we had collected, you can get that data. Every seed collection we make gets a unique number. It's called a seed accession number. And that number travels with that collection throughout every part of at the nursery. So when it's in propagation, when it's out in the nursery yard, that seed accession number will always tie back to all that original information from the seed collection. We kind of call it the passport papers. It always travels with it. And when are we collecting? We're collecting almost all year long. So our collections will start in April, and we can collect we're usually finishing up collections at the beginning of December, and then sometimes, we're out there in January, February, making a couple collections as well. But we're doing this all year round. We're collecting, from all these different ecosystems and habitats that make up our flora. We're collecting from all the different layers of the habitat, so the forest floor to the canopy. So ferns, grasses, herbs, shrubs, trees, everything comes to seed at different times throughout the year, and it's what we're constantly having to track and prepare for. So we're always preparing for seed collection. We're always, tracking what's going on with the species out in the field. We're making the collections, and then we're also cleaning all of the collections throughout the year as well. And our priorities are usually project driven, and this changes throughout the years. After Hurricane Sandy, we had a lot of coastal projects. I was spending a lot of time, out there in the coastal habitats collecting, because that's where restoration was happening. So it's really following the trends of what what the city is involved in. The trends of the greater industry, pollinator gardens are huge. We work with a lot of community gardens, and we're constantly growing tons of species every year for pollinators. So really that determines our collection priorities for the year. And we have a huge priority list, and we're identifying all of our target species. We have tons of data on where our potential collections might be. And then really it's tracking the phenology, because this changes from year to year. And this is the most important part, because as a seed collector, if you're out in the field and you don't collect seed, you kind of feel like you failed at your job if you're not there at the right time. So we really need to understand, when something is flowering, what's that window, what might be affecting a flowering period or a seed production period. And then all of these clues that nature gives us on when is the seed actually ready. So we have changes in fruit color, and this is something that is pretty probably the easiest one to see. When animals want to eat a fruit, when we want to eat a fruit, it's when it's nice and blue for blueberries or red for strawberries. It's really in that change in fruit color when you know that seed is ready. Because if it can be digested, then the seed is ready to be deposited elsewhere. So that change in fruit color is really important as an indicator of when seed is ready. Changes in the seed coat or cover color. So this will always be a really good indicator. And sometimes you can peek under a seed coat to see if indeed that seed has started to change color itself. So, in the bottom left-hand corner, you have the American hazelnut. You can, pry those seed coats open and check to see if that seed has started to turn brown. It's sometimes getting to the seed before it drops. So you have to really maybe even take it a little early before that seed coat cover changes. And then fruits breaking open is always a good indicator that the seed is ready. It's getting ready to naturally disperse itself. So if you start to see some of that happen throughout the population, you could probably, be able to make a collection on that day. On the bottom right-hand corner, that's hibiscus machutos, our native hibiscus, and that matures at different times, right? Flowers are opening at different times, so you're going to have seed ready before the next ones are ready. But that's great, because now you're really able to just take a portion of the population, and seed will still be ready later on for wildlife and the natural seed bank. And then our fluffy species are really easy to understand when they're ready, whether it be the populus, the aspens that are flying through the air, all your aster species once they start fluffing out, our milkweeds, and the little blue stem at the bottom. When that seed is getting ready to fly, it is definitely mature and ready for collection. And then you can always do a cut test. Doing a cut test out in the field is really easy, as long as you have a knife and you can just cut open that seed and understand, or clippers. You can cut a nut open and know if it's full or not. And this is really important with a lot of our tree species. A lot of them will abort a certain amount of seed early. So you will have to make sure that if you're all of a sudden finding nuts on the ground, you cut a few open and you're like, oh no, those are empty. This is just the first initial abort. I have a number of weeks. So the cut test is always really good to do. And then we have some general rules on seed ripening. So a lot of our annual and herbaceous perennials will be ready to collect seed from 2 to 6 weeks after bloom. Every species is unique. But, if you have a kind of idea, you can start watching these species and understand, when they're going to be ready. Because we have so many years of data of seed collection, we have very particular windows. Some of them might be 2 weeks, 3 days. And of course that will like change, with the weather that particular year, but it's pretty spot on. So, we know when these seeds should probably be ready. And a lot of perennials have a longer window of opportunity for seed collection, and that's because a lot of the seed is going to be maturing at different times. So you're not necessarily going to have it's not like our spring ephemerals that will just, have a very quick seed maturation before the plants senesce. Indeterminate inflorescence, so your spikes, racemes, panicles, umbels, all those species will ripen at different times. So if it flowers at the bottom first, those seed pods will be ready before the ones up at the top. So you might be picking like just a few seed pods off the bottom. What's great about our graminoids, our sedges, and grasses, you can collect a little early. Collecting with stem, there's enough energy in that stem for the seed to after ripen. So we're able to have a bigger window on that as well. And this is always an interesting one to remember, is that populations north of NYC mature later in the spring. It's going to be colder up there. And then earlier in the fall, because it will be colder up there earlier than it is in the south. So you have to kind of flip that when you're traveling either north or south. And then always the weather is dependent on seed. We had a terrible drought last year. We missed a lot of species did not set seed. And that's just part of the game. So really the weather really makes a huge difference on what is going to set seed, if it's going to have a great, and some species do great in drought and others do not. So it's really really knowing those species intimately and how the weather may affect it. And then every species is going to be unique in the amount of seed it produces. Seed production can be cyclical. A lot of our tree species have masting years. So this past year, there was barely any acorns. It was not a good year, not only probably because of the drought, but also because it was just part of the cycle that it's not a mast year. When it is a mast year, I am on my hands and knees picking up acorns all fall. My back is broken by the end of the season. So you just got to get them when you can. And then there's the poor seeders. A lot of your grasses, a lot of things that are wind pollinated don't produce a lot of seed. So you have to make a massive collection to be able to get even a small amount of seed. The species on the right is the beach grass, Ammophila breviligulata. This spreads by rhizomes. It doesn't have to produce a lot of seed, and it's wind pollinated. So it's just a poor seeder. And if you know that you have to make a large collection if you want to get any amount of seed for production. And then the weather, of course, and then disturbance is a huge one. And whether that be natural disturbances or man-made. So we will scout out a population, we will think this is going to be so great, we're going to go back in the fall and collect it, and we go back and it's been mowed, the whole field. Or a fire went through the area and everything is gone. So disturbance can completely change the seed production. And then predation as well. A lot of insects and animals will completely wipe out our seed source on some populations. So that's always something to keep in mind as well. And when we bring people out in the field with us to collect, they're a little bit nervous. They're like, they don't want to hurt the plant. But you know, we're out there and we're just stripping and plucking and snipping and shaking and doing all the things that we can do to get the seed. And really you're just using, it's a lot of handwork. We do have some tools that we bring out with us, pruners, pole saws, or poles with hooks on them to pull branches down. So really we're trying to get the seed in any way that we can. And, you know, we do have, you need to know, of course, what you're collecting from. Some Carrick species are, you know, they're set apart by micromillimeters on particular plant parts. So having a hand lens out with you in the field, or plant manuals or floras, if you need to make sure that you're collecting the right species, is always important as well. At our home base, we are laying everything out in the seed lab. It's really important when you get back with a seed collection that you're letting it kind of dry. The excess moisture is kind of drying off. All the creatures and spiders and bugs that you by accident collected are crawling out. So we're leaving our seed collections out in trays with just some newspaper underneath them to collect any of the seed that falls once they're drying. And we'll probably leave them out for about 2 weeks before we package them up, and then put them in an area in our seed bank before they get completely cleaned down. And labeling is also so important when you're doing seed collection. You may think, I'm going to remember what this is, or I'm going to remember when I got it, but you will never remember. And you have to label always inside the bag, outside the bag, in case the tag falls off the bag, there's the tag inside the bag. All of those things you need to remember to do. And our labels just include the species name, the location where we collected it, the date, and then that unique number that we give it. And then the seed processing and cleaning is a huge part of it as well. Once we get all the seed, you know, we have to clean it down to its purest form as possible. And this is labor intensive. A lot of it is done by hand, and it can be replicated very easily. You know, we do have machines for large collections, but you know, this right in front of you in this photo is stair tread that you can buy at a hardware store. And this is what we use to start rubbing our seed down. And this is removing a lot of the seed from the actual plant parts. We're rubbing off the seed coat. So this is just using friction on a rubber mat. You can also do this with screens. A lot of screening happens with our seed. So again, you're just rubbing the seed off of that plant material, or, you know, hand picking it. And then we do have certain machines. These are called clippers. And they will clip out the seed. So at the bottom there in that tray, that's all the seed that falls. It's a machine that has fans and belts that will force air through. And there's different settings for each particular species and screens. So there are machines that can be used, but a lot of seed cleaning can be done by hand. And this is a larger version of the clipper. So these are the machines that are used from our fields, our seed increase plots. And then everybody always, you know, loves to know about our fleshy fruits. You know, we are cleaning by hand. We're picking off all the stem material, and then we're just using a blender to clean the fleshy fruits. It is a blender that is altered. We file down the blades on the blender, and then you can tape them with electrical tape, or you can get that like plasti dip that you can dip them into and create a plastic coating over the blades. And that's just to ensure that you're not nicking or breaking the seed when you're blending it. All you want to do is get off that fleshy fruit coat. And what's great is then you pour it into a bowl of water, you swish it around, all the pulp, since it's so light, will float, and all the seed will sink. The full seed with an embryo would sink. A lot of the empty seed will float, and then you just pour off the top, and then you have all the seed at the bottom. And some species are a little tougher. species like our eastern prickly pear cactus, you get these beautiful fruits once they're ready, but those fruits are filled with a jelly that is so thick. And you scrape the seeds out of the pods and you have this thick jelly around all these seeds, and we had to figure out how to get it off. So we soaked it for a while and it just became a little less jelly. And then we would let it dry out. We found putting it on our greenhouse benches that have bottom heat helped with the drying process because it was taking so long for the shell to dry. And then we were eventually able to just rub off all that dry gel. So sometimes it's a very laborious process, but you get down to that pure seed and it's ready for germination. And there's so many resources out there on how to germinate native seeds. We use a lot of different resources and we use a lot of our own trial and error data. We've been producing plants since 1995, so we have a lot of data ourselves. Hopefully one day our amazing propagator will be able to publish some of it. But it is available. We do share our data and we love to help, other propagators figure out or, use our methods. But really some people have never tried to grow these plants from seed in a greenhouse setting. So, we are working from scratch on a lot of them. There's not a lot of data out there. And some species are so difficult. They have double dormancies, things that we can't even begin to understand. And just putting them out in a tray to go through the seasons year after year, we will get better germination doing that than trying to, do any fancy germination techniques, in a refrigerator or whatnot. So sometimes just putting it out there is the best way as well. And all of this data, has helped inform so much about our native flora here in the 5 boroughs. And I always love to plug this document. We put a lot of love into this document. It's a native species planting guide for New York City. It's a lot of guidance and information about our species and how they can be used in the landscape. It's available online as a PDF. It was originally done in 1993. The second edition came out in 2015, and then the third in 2019. We are obligated by local law 11 to revise this every 5 years. So a new revision will be coming out soon, but it's so much great information on the species that are going to be really effective in the landscape here in New York City, that are tough and can survive, and all the different ways that you can, potentially use them. So I hope that you seek this out if you're really looking to understand, what native flora can do and how it can function in the landscape. And I think that's it for me.
Awesome. Thank you so much. There was lots of information in there. I know I learned a lot. We do have some people throwing questions in here. I did put a link to that native species planting guide into the chat. So if people want to check it out. All right, let's scroll back up here. All right, so first question from Frank. He says, you said sold. So are these plants being sold to the public, or are they only for certain kinds of organizations, or what?
Right. That is a loaded question. As all of our plants are for first and foremost New York City public spaces. So we are working with people who are doing projects on New York City public land. And we're also, we do work with some different groups that like different landscape architecture groups and whatnot that are doing, green roof projects. We've been working with the people that are, the DEP and other people that are doing, a lot of our stormwater basins and catchments. And we try to work with school groups and community groups that are looking to do native plantings around the schoolyards or their community gardens. So we try to work with as many people as we can. Our material is, though, committed first and foremost to the projects that we are growing for. We are also just trying to get these plants into the landscape as much as we can. So yeah.
Awesome.
Technically not open to the public.
Right, but certain members of the public that are involved in certain projects could be.
Right.
Is there also a facility in the Bronx? I know I've heard about a growing facility for parks up here, but I'm not sure if it's
so that's
like
the
kind of like a
citywide.
satellite.
That's the citywide nursery. They do get some of their starter material from us. They provide a lot of the horticultural material to the 5 boroughs for the different hort programs. But more and more people are wanting native plants in the horticultural settings. So we do funnel a lot of our starter material to them.
Awesome. All right. Jeanette, you have your hand raised if you want to go ahead and unmute yourself.
Thank you. That was a really very good presentation. Thank you. I have a lot of questions, but I know that I don't have time to pop all of them. I have one related to climate change. That's something that I think is very into. And you mentioned that you guys have been collecting information from 1995. It is any way that you guys can overlap all this information about the plants distribution and climate change? That's one. The other one is like, we also have a lot of plants that are not native, no? But you guys are trying to restore by putting these native plants. And what about a project, or do you guys have a similar project that you are putting out all these non-native plants in order to create more space for the native plants to repopulate these areas? Another one that I have, and you kind of said a little bit, is about these things that you're working with kids. I think that one for me, it's something very special because I think they are the next generation, yeah. And if we really already empower them with information, they are going to be able to make them better decisions that we have been doing. And I don't know if you guys do workshops for kids, and if not, I will be very interested to know more and see how we in the D3 group, we are in the Department of Education Green Group, how we can do something and partner with the teachers to really start teaching actively something to the kids about these things, the seeds. I think the seeds are fantastic. Thank you for the presentation.
Yeah. So for the first question, we really need a data analyst. We actually had a whole conversation about it today. But yeah, we have all this data. And I think, I think it does, I think we are an integral part of that conversation because, I think so many people are ready to jump to assisted migration and let's just like move all the southern species up north and, plant for the future where, we have a more at least I don't want to speak for, my department, but, I believe that urban spaces are that, what we should be looking at. We should be, looking like our temperatures and, what our habitats go through, they're so much more extreme than just even the outside towns and cities and regions. So what's happening within the 5 boroughs is kind of a great window into what's going to happen in the future in the outlying areas. And, yeah, we don't, a lot of our flora has been lost from New York City, and there are species that we don't have the right habitat anymore for those species to exist. But there are so many species that still do exist and have been adapting, and that's what we should be looking at. As for your next question, we're involved in so many projects within the Parks Department where they're doing large scale, invasive removal and restoration, and they're using our seed and our plants to be planted after that treatment is being done. So we're constantly working with those groups on what species will be, those really important pioneer species, aggressive, that will cover the landscape, that will hopefully compete with the invasive species. And that's an important part of it as well, because everybody knows about the Million Trees Project, and it's important to plant trees and plan for that climax habitat. But really those pioneer species that are going to do the work on the ground first are important as well. And that's where your grasses and your raspberries and blackberries and all of those species that really are your pioneers that come in first, those are plants that we provide. Those are seeds that we can provide for those tough areas. And then for the last question, we would love to have the capacity to do so much more in education. We are limited, our staff time is limited to, what we can do. Currently, we hold a tour once a month at our facility where we do have school groups come in. We have set up different things with school groups where, they'll come in for like a work day, and they can learn about everything that we have going on at the nursery. And I think it's always our hope that we can do more with education, and if we had an education coordinator, that would be great too. But I think that yes, education is for sure, the route to go, and we try to offer as many internships as we can, and we try to, work with as many groups as we possibly can.
Thank you for your answers, Heather. Okay, so next up in the chat, we have Donna, who asked, do you do viability testing on seed lots?
So I've been doing a project for the last couple years now. We are not actively doing viability testing on any of our wild collections besides this project for the last 2 years. Our seed increase program, they actually send their seed out for testing because it's a seed product that is being sold as a seed mix. So they have their PLS that they're required to have on that. But for our wild collections, we just started doing a seed longevity project where we're sowing very, very old collections to see, what still is viable. And, we're going to be collecting data from that. We have found some, even, the first year, it was fascinating to see some of the species that germinated. We have this rare Pinus echinata. We had seed that was almost 20 years old and it germinated. And it was not even kept in the best conditions. So we hope to do more. We do have a germination chamber on site. We were doing some testing with that a number of years ago, but we also don't have the staff that can be dedicated to those viability tests.
I wonder if that would be something that could be also like student type projects.
Yeah. For
because
sure.
because I'm doing my PhD right now, I'm always thinking about those kind of things of what would make it good, even like master's type projects.
Yes. Yes, we have like a vault of, I feel like information and data and things that can be mined at our facility. And they would be fantastic projects.
I think it's interesting because you've mentioned a lot of kind of, if we had more people, or we had these certain kind of positions, and it is like, because the Parks Department as a whole has kind of like a certain function that's not necessarily like primarily academic, let's say, that kind of falls to the wayside. But meanwhile, there's tons and tons of really like great information that's like internal. So it's
Yeah.
good to have some of these, like be able to release some of these guides and have like some of that information start to get out there.
Yeah,
Totally.
for sure.
And that's why we like to have these kind of talks also, to like let people know what's going on, that they can start to then see how they can kind of get involved in things.
Yeah.
Got lots of questions in here, along with lots of nice comments. Steve Young asks, do you not plant in areas that you know are going to be hammered by deer?
We are more selectively doing that now. And more and more deer fencing is being written into contracts, or extra money for tree restitution or whatnot is going to deer fencing. So whether it's fencing the actual plantings themselves, or an area in a park that is indeed going to be planted, or fencing some of our existing populations that are vulnerable. We have like kind of a wish list of areas throughout the 5 boroughs, in particular more rare plant populations that we have been fencing and will always continue to push to be fenced. So that is a real issue for sure. And it is definitely more part of the conversation now, that we're not going to provide our material to some projects in Staten Island and the Bronx if there's not deer fencing included, particular species.
And I saw in that that list that there is stuff about like species that are less popular with deer, for example.
Least preferred.
Yeah, well, he's right, least preferred, because they'll probably eat anything.
Yeah.
Heather, I have a question for you actually. So do you, when creating the priority lists, or thinking about planting these species into new green spaces, do you take into human desirability and cultural desirability when thinking about that? Like, I can think of poison ivy as a great native plant that supports a lot of native pollinators and like really interesting insect species, but it's less than desirable for humans. Do you kind of take those things into account?
Yes, but we are always trying to push new species to be used that people are not using, but they're fantastic. Not poison ivy is not one of them, but that one's doing just fine. But yeah, I think that's what's a huge part of our job too, is working in the early stages with some of these project planners to really flesh out a species list for these plantings. Because to be honest, I mean, we usually get the same palette, right? It's a coastal project, they want the same species, and we're always trying to push diversity into these palettes, because, if we have the material to do it. And I think it might be a shift, in the Parks Department in the future, where, these large scale plantings that are being done, they are, 2 years under contract after planting with the contractor. And then once that contract's over, it comes back to us to manage. And part of that management is to start to increase the diversity of the plant species that are in a restoration. And that's where we can go back and say, okay, what plants can we grow that, aren't in this basic palette, that can be a real benefit, not only to the habitat itself, but, the insects and animals and whatnot that we're trying to bring back into this habitat.
Awesome. Yeah, that's really cool to consider, like the ecological implications of certain species that you're trying to consider. Yeah, that's awesome. Great. Okay, so Tammy asked in the chat, she said, you mentioned that you collect the seed from 3 states within 100 miles of circle. Do you share these seeds with other states that do similar public service projects?
So every permit that we get from these entities, it's part of the permit process that because we're a non-for-profit nursery, we can't give plants back to these entities that permit us to, collect on their lands. We do offer, though, that they have access to any of the seed in the seed bank that came from their lands, if they indeed want it back to propagate themselves or use in particular projects. So that is always an offer that's on the table. It's kind of our good faith in our permitting process that, you allow us to do this and come and collect on your lands, and you always have access to that seed. The only other way that the seed is going outside of the city is if we are giving the seed or starter material to nurseries that are then the city is buying back from for projects. There is the Mid-Atlantic Regional Seed Bank, which, they're more so serving the greater region. The collecting that we do is indeed meant for the 5 boroughs.
Thanks. Okay, we got a couple other ones in here. I scroll back up. So Marsha had a question about cutting into the seeds that I have a kind of piggyback question off of that. So she asked, when you cut the seed, what are you looking for the most amount of the white part, and is that the endosperm? And my kind of follow-up question to that is, how long are these seeds then viable? And I guess it really depends on the species, but.
Yeah. So when we do the cut test, a lot of the times we are looking to make sure that embryo is full, right? It's filling the entire seed cavity. And we are then making sure there's no insect damage. So a lot of times if you cut open a seed, you can see like a black hole where maybe an insect has started to eat the embryo. So or it's just underdeveloped, right? Like sometimes you'll cut open a seed and it will just be halfway full. And that means that it's either underdeveloped or you came too early. So you are looking for that full white embryo. And then each seed, each species or, group of species is different. We have recalcitrant seed, which can not be stored. And then we have orthodox seed, which can be stored. So your recalcitrant seed is seed that, it's usually your very large tree nuts and whatnot that have a high oil content or high water content. They can't be dried down. If they're dried down, they die. So those seeds, we can't store in our seed bank in a normal way. We can maybe hold them in our cold storage for maybe a year, but they have to be, directly planted usually. And that changes, with plant families and whatnot.
Nice, because I know here at the garden we have like a seed collection I think was donated, but people, and I show it to people sometimes, they always ask it. I'm like, these seeds are so old, they're really just, they wouldn't, and none of them would be viable at this point, but they still have, like use for like anatomical studies and stuff like that. But I know that's something that might not be like well known to people, like how long the seeds are viable.
It depends on how they've been stored as well. So, I was actually shocked with some of, this project that I've been doing. I was shocked at some of the seeds that germinated. For example, the Agritina altissima, the what's the common name?
What?
Boneset. It's just the common, Eupatorium species that is in the landscape. And in the Asteraceae, usually a rule of thumb is that the Asteraceae family, the seed is not viable for that long, maybe like 5 years or so. And then the viability drops off, really far. But this seed that I sowed, that was stored in a plastic bag was, fluctuating in temperatures here and there. It germinated and it was, almost 20 years old. So you all of a sudden have this, outlier that's like, wait a second, how did that happen? But it's one of those really tough pioneer species, right? It will just like come up everywhere and just like be in the toughest spots ever. So it's species by species.
On the topic of germination, Frank asked a little bit earlier in the chat. He said germination of uncommon species is often most challenging. Do you publish your discovered germination protocols?
We have not done any publishing of our protocols. We hope that might be something that we can do in the future. Again, I think it's just lack of time that we've been able to, do that. But again, a fantastic data mining project. But yeah, we definitely have found some cool ways to germinate different species. And we're constantly refining those and trying new things. So I think hopefully that could be a reality someday.
Are there any pyro or fire dependent germination seeds that are common in the Northeast or in New York here?
Yeah, I mean, a lot of your pine barren species, the way that we the Pinus rigida, which is your pitch pine, we actually bake those cones in the oven to have them open up and release the seed. So they're very serotinous and they're closed up super tight. We heat them to a high, we put them in the oven at like 400, 500 degrees and they pop open, and we're able to pick all the seeds out. So, there are species that require fire. We had one propagator for a while that would light some, they would take pine needles and, mix it in with some seeds and then light it on fire. So we have done that before.
That's very cool. I keep having to scroll back up and down at these things. Well, one question that just came in from Allison, have you collected or propagated Impatiens capensis, and is that an easy or difficult species to work with?
We have collected it before. We've never tried to propagate it. It actually went into a seed mix that's going to be put out into the landscape. It's an annual. It's rare if we're propagating annuals, unless it's for like a project that we know that's going to be on time to get the plant out there in spring so it can go through its life cycle. But a lot of our annuals get put into seed mixes that are going to just be put out there in landscape. More people are coming to us for seed balls or seed bombs, and we try to put annuals in those. So we've never tried to actually grow that one.
It does seem like Frank might be our resident expert on the growing, because he has some comments about it.
A little bit. Oh, go ahead.
Oh no, go ahead.
Oh, I was going to ask a new question or read one. Yeah. Okay, cool. So a little bit further up, Caitlin asked, and she's kind of talking about, well, it's interesting thinking about introduced species interacting with the native species. And she said one of your later slides mentioned that selecting native plants that can resist these invasive species. But I'm wondering if you do collect seeds from or cross-pollinate with any introduced species. It struck me that the landscapes of trash and concrete are introduced landscapes that the native plants you work with have adapted to. By the same token, some introduced species have self-selected to be adaptive in a landscape introduced to them. Also, thank you so much, or thanks for such glorious presentation.
Oh, could you summarize the question for me quick?
I guess she is just asking about if you collect any species that are introduced as they are sort of self-selecting and kind of introducing themselves into this landscape, and they seem to be working well, even though they're introduced and maybe displacing some native.
Right. We are not collecting naturalized species. Our idea of native is conservative. We are really looking at native flora of species that have been here, kind of, since since, you know, we have the records for, you know, if they've been used traditionally by Native Americans, any plant records that we have, you know, so we're not collecting anything that is considered naturalized. The New York Flora Atlas is a fantastic resource that really has that information for every species. And if you look up a species, you know, they even have some undecided on them. So I think that we're always going to stay, you know, on the way more conservative end of things. And we're not going to be in that arena.
And I would just to maybe kind of piggyback off of that, the thing with I guess the non-natives that are self-selecting to do better in this environment are in fact the kind of thing that you don't want, I guess, from the perspective of them kind of displacing the native plants. That is kind of what makes them invasive in nature, because you can even have like invasive natives where they're just like gonna grow at the expense of other plants. I mean, that's how I kind of, I know that definition has a lot of different meanings to different people.
Right. And, you know, we collect species that people don't want to put in a built landscape because they are bullies, right? They're going to potentially take over. But those species are important in some scenarios. So I think it really depends on what, you know, the project is intended for. So if you're just looking to have something compete with, you know, mugwort or frag, those aggressive species are important.
Yeah. Okay, there's one or two more questions here and then we'll wrap it up. One thing, well, I have one question first. So you mentioned when you're doing the collections, you want to make sure that it's a large enough population, that there's over 250, and that you're sampling about 50 of them. How does that kind of complicate when you want to collect seeds for something that's much rarer than that their population sizes are not that big, or there aren't many populations, but so that balance between you want to have some of the seeds to do, but you also don't want to be taking too many of the seeds.
Right. Rare plant collection is a whole other ball game. And there is a whole different protocol for doing rare seed collecting of rare plants. There's a whole completely different protocol.
Okay.
And, you know, for a lot of the, you know, the rarest of rare plants, you know, there's the Center for Plant Conservation, there's the CPC holders of particular species where they are, they've taken the responsibility of doing those collections. The only collections that we've been involved with really on that level with CPC has been the sea beach amaranth. The sea beach amaranth, you know, is endangered across the entire eastern seaboard. We had a really healthy, amazing population in the Rockaways for many years. It has fluctuated here and there. It's actually on the low end of numbers. After Sandy, it skyrocketed. I think we had some of the best numbers along the eastern seaboard. And, you know, it's a whole different type of collecting when you're doing rare plants. But as you know, the largest landholder in New York City, the Parks Department, you know, we do have rare species on our lands, and it is up to us to help conserve them. And we are doing some of that collecting, and we're even doing some, you know, plant rescues, you know, where we're digging plants up because of a project that's going to be destroying them. And trying to move them elsewhere and relocate, or bringing them to the nursery to pot up and be able to collect seed from. So, you know, that's a whole different thing and not really what we focus on. We do some work. There is a conservation, you know, arm of our program, but we really are focusing more on those common species.
Because those probably have a higher chance of kind of success in the first place. Awesome. Okay, so the last thing it looks like we have on here, well, Nadia asked, what's the most unusual germination protocol you have which I think would be a good thing to end on. But before that, people would love to hear some aggressive species that could compete against mugwort and Phragmites.
Okay, so aggressive species. I think there are some solidago species that would be fantastic. You know, the Canada goldenrod, Solidago canadensis, it might be the only plant that grows there for a while. Sometimes you see a field of it and it's the only species, but it's a fantastic pollinator plant. There are, you know, we're going to be doing a lot of, the Parks Department is about to embark on a lot of wetland restoration. I believe there's a big grant that was awarded to do a lot of frag removal. And we are coming up now with some species lists of what's going to go in place after they do the frag treatment. But it's pretty fantastic to just kind of take clues from nature. If you've ever crashed through a frag field, which wear long sleeves if you do, you will find the species that still grow there. You know, and there was this fascinating part of Staten Island down at Oakwood Beach where, you know, people that lived, you know, in these little bungalows that all were destroyed during Hurricane Sandy. But when they lived there, they would constantly mow the Phragmites, and the Shenoplectus americanus all came back, and it was just there in the seed bank, and it was a full field of it in this place that they constantly mowed every year. So I think taking the clues from the landscape is important, of the species that you see that pop back. Calamagrostis canadensis has been an amazing species to compete with frag. Tripsicum dactyloides is a good frag competing species. Anything with that like rhizome network is going to be important. There, Oakwood Beach again, there's, you know, this one area where it's a field of every Osmunda species, the fern species, that is basically keeping the frag at bay. It's kind of wild. The Hierocloe odorata, the sweetgrass, that still grows in the 5 boroughs, and I've seen that, you know, compete with the frag. So I think, you know, spending time in these spaces and really, you know, understanding these species is the best kind of mirror on how to do the work.
Awesome. Well, it's always good to hear that there are still, you know, it's not that you said it was all doom and gloom, but it's easy to feel like it is sometimes, that there are still kind of like places that have these like healthy ecosystems going, that it shows that it's not, you know, we can reclaim some of these spaces.
Yeah.
And blow torches, but
yeah, as much as it's heartbreaking sometimes to, you know, hike around these areas and see the trash and the disregard, and, you know, the Economic Development Corporation building another building somewhere, like, then you see, you know, the highlights of, you know, these species that have still figured it out and will keep growing alongside of it all. And I wish I could remember a really ridiculous propagation technique, but I'm not the propagator.
fair enough.
But I must say that one of our seed collectors did collect bear poop and wanted to, you know, grow what he found in there. So you could always do that.
That's very cool. I just wanted to read out a comment that was in here just for people to know. Lucy said the general public is welcome to sign up for tours and gardening events on the NYC Park Stewards webpage if they're interested in seeing the nursery. So there are some, looks like some kind of ways to not only see the stuff, but to get involved with stewardship and helping out.
Yeah,
So-
and we've been working more with the stewardship program. We're even hoping to have like, last year we had some collection days where people could come out and do collecting with us for big scale collection, like in the marshes. If it's a mast year, you know, collecting for acorns. So yeah, definitely keep an eye out.
Nice. And maybe that's even something we could try and set up an EcoFlora work day or trip to do. That would be really cool.
That'd be awesome.
Awesome. Well, thanks everyone for coming. Thanks Heather for a great presentation and everyone for your great questions and comments. So one more time, I'm going to throw the link to our mailing list if you're not already on it, because we have lots more exciting talks and field trips coming up. So stay tuned.
And if you're in New York City, Lydia mentioned we found 38 species of winged insects in New York City so far this month, but there's lots listed that have been observed before. So keep looking for those pollinators and plant associated insects. And thanks, Heather, so much.
Thank you.
Have a good night, everybody.
Wonderful talk. Thank you.
Thank you so much.
Thank you.
It was great. Thank you.
Thank you. Fantastic.